Hello, my name is Joshua Mugabi. I welcome you to topic 11.1, Inheritance. Learning outcome A. Understand the process of cell division and significance of meiosis. We are going to look at learning activity, explain verbally and in notes the significance of meiotic cell division in maintaining the chromosome number. Item 1. Significance of meiosis. We are going to look at maintaining chromosome number. During a biology lesson, learners discuss why humans do not end up with double the number of chromosomes in every generation. Why do humans do not end up with double the number of chromosomes in every generation? Here we have our diagram showing haploid male gamete with 23 chromosomes plus a haploid female gamete, 23 chromosomes, and we get a diploid zygote with a total of 46 chromosomes. Our tasks: state the significance of meiosis in maintaining chromosome number in humans. We want to state the significance of meiosis in maintaining the chromosome number in humans. Now, meiosis maintains a constant chromosome number in humans across generation. So what we are saying here is that the number of chromosomes in humans from one generation to another is maintained the same. For example, if you look here, this is our zygote. The new generation will have 46 chromosomes from generation to generation. But what brings that is meiosis, is the one that maintains a constant chromosome number. How? For example, if you look here we have a haploid female gamete or male gamete, the sperm, and it has got 23 chromosomes. And here we have a haploid female gamete with 23 chromosomes. The formation of gamete is by the process of meiosis, and we end up having 4 haploid daughter cells, each with a half the number of chromosomes. So the gametes that are formed, the sperm and the female gametes, they have half a number of the chromosomes, such that when the male gamete combines with the female gametes, the number sums up and it gives us the normal number, which is 46 chromosomes. So we can see that it is meiosis which is maintaining the constant chromosome number in humans from generation to generation. Explain how meiosis achieves this. We want now to explain how meiosis achieves this. Achieving what? The maintenance of the chromosome numbers. Here we have our diagram here. Let's first look at it. When you look at the beginning here, this is during interphase. Interphase here, we are seeing the chromosomes here, and the chromosomes are duplicating to form sister chromatids. So here we are having two pairs. 1, 2 from maybe the paternal, 1, 2 from the maternal, representing the 23 pairs. We are using 2 pairs. During interphase, they duplicate. Each chromosome duplicates to form sister chromatids. So this one here will duplicate and give us these ones. Now, this is one chromosome. This is one chromosome. After duplication, it forms what we call chromatids, sister chromatids. So all of them will duplicate. Now, after that one, there will be alignment of homologous chromosomes. Chromosomes that are of the same character, they will come together. Like the way we are saying homologous chromosome, this one and this one, they are homologous. Therefore, this one and this one, they are homologous chromosomes. They will align themselves. And this is during meiosis 1. And in meiosis 1, what is going to happen? The homologous chromosomes are going to separate. And when they separate, we shall end up with 2 cells. And when you have 2 cells, if you remember very fast, in anaphase 1, it is the homologous chromosomes separating. This is homologous to this one. So when they separate, they will go in different cells. And when you look at it, you can see that this one has gone to this cell. And you will see that this one has moved to this cell. So the homologous chromosomes have separated. Then when we go to meiosis 2, now we shall have the sister chromatids separating themselves and going to different cells. So we are going to end up with 4 daughter cells here. If you look at it, 1 cell, 2, 3, 4. But when you look at this chromosome here, let us just look at 1 and emphasize on this one. Let's look at this chromosome here. So when it is going to separate, it will separate in the middle. When it separates in the middle like that, then the sister chromatids have separated. Now, this sister chromatid, this side, this one will go in this cell. And this sister chromatid, this side, will now come in this cell. So you can see that in meiosis 2, it is the sister chromatid separating. Whereas when we were in meiosis 1, it was the homologous chromosomes separating. So we are asking ourselves a question. Explain how meiosis achieves this. So let us look at that one. Here we say that meiosis reduces the chromosome number from a diploid, which we call 2N, and that is 46 chromosome, to a haploid, which is N23 chromosome during gamete formation. So when you look here we had a total of 46 chromosomes. They underwent meiosis, and when they went meiosis 1, now they became haploid, half the number, because this is one cell. And when one cell divides into two cells, meaning that a half will go here and a half will go here. Now what was a diploid, 2N, now this one becomes N and this one becomes N. Now during meiosis 2, it is the chromatids which are separating, so the number remains the same. So we shall form 4 daughter cells, each one with a haploid number N, 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 because remember the diploid here, we had duplicated the chromosome. So when the sister chromatid separates and they divide and go into 2 cells, the number of chromosomes remains the same. So here, after meiosis 1, we have haploid. And after meiosis 2, we still have haploid, but 4 daughter cells which are haploid. Now, because of the changes that are taking place, now when you look at these different cells, you will see that they appear differently. If you look at them, the chromosome, look at this cell here. And I want you also to look at this next one. When you look at those chromosomes there, look at this chromosome, it has got a part of the paternal and part of the maternal attached. This one is only from paternal. When you look at this one has only one side. Okay? Maternal fully. And here there is maternal attached to on some part of paternal, which we called crossing over. So when you look at all these cells here, you're going to see that there is going to be variation. So because of those different chromosomes going to different cells, and what happened there was crossing over, which we said it takes place in anaphase 1. So when you look at them, they are very different chromosomes. So the gametes are going to be different, and also the results that we are going to get out of them are going to be different. And that is what is going to bring about variation, which we have talked about. But this time we are talking about haploid, maintaining the chromosome. Now what we have formed here is a gamete. This is a gamete, this is a gamete, this is a gamete. And this is the parent. Now when you look at the parent, I want us to look here very carefully here at the parent. We have 4 chromosomes representing the 46, but here we are having 4 in the parent. Now let us go to the gamete and we see. If we pick one gamete, this one, and we compare it with this one. Let's compare, and you see this one has got 1, 2, 3, 4 chromosomes. Let's come to the daughter cell. The daughter cell has got 1, 2. So this one is a half the number of chromosomes compared to the parent. This one had 46, which we call the diploid number. And it goes to haploid number, which is N23, which is a half the diploid number. So the same here, this is diploid and this is haploid. This is a half the original number. So that is meiosis doing that one. And we saw earlier that when the female gamete combines with the male gamete, again now they go back to diploid number. So still here, we find that during fertilization, fusion of the 2 haploid gametes restores the diploid number. So the other side we were looking at meiosis taking place, and having a diploid turning into a haploid. Now, when we come here, we are now seeing from meiosis to a new individual, making it diploid. So when you look here, you will see that this one is a haploid female gamete. And here we have a haploid male gamete. If you look at the number of chromosomes, you will see that this one has got 23 chromosomes. And you see that this one also has got 23 chromosomes. So the 23 plus the 23, they are going to sum up. And when they sum up, we are going to get what we call a diploid zygote, that is during fertilization. So this 23 plus this 23, we are going to get 46 chromosomes. This is a gamete, this is a gamete. When they combine together in the process called fertilization, we form a zygote, and the zygote now has got 23 chromosome plus 23 chromosome of the two gametes to make the 46 chromosome. So we are saying here that during fertilization, fusion of the two haploid gametes, haploid means a half, which is 23, restores the diploid number. Diploid is 23 plus 23. When you add them, you are going to get a diploid number, which is 46. Then this prevents chromosome numbers from doubling in each generation. So it is meiosis which results into halving the number during gamete formation, and when the gamete come together, half, it goes back to a diploid number. So meiosis prevents the number from doubling from one generation to another. Item 2. We are still on the significance of meiosis, importance of meiosis. Now let us look at plant breeding situation. A plant breeder crosses two maize plants, each with 20 chromosomes. The offspring also has 20 chromosomes. So here, the breeder is asking himself, he's breeding two maize crosses, and each maize plant has got 20 chromosomes, and this one 20 chromosomes. And then the offsprings that result, they also have 20 chromosomes. So let's look at that task. Identify the process responsible for maintaining the chromosome number in this cross. They want us to identify that process that brings about the maintaining of the chromosome number in this cross. If this had 20 and it was crossed by this one of 20 chromosomes, and then we got these ones also with the 20 crossed with 20, we would expect that one to give us these ones having 40, but they also remain with the 20. So which process results in these ones being 20? Yet they are coming from this one and this one, which are both 20. So in a layman's language, you would say this is 20 plus the 20 will give us 40. But here they are saying that the offspring also remain with what? 20 chromosomes. So which process is that one that maintains the chromosome number in this cross? That process is meiosis. The process is the meiosis. Explain the significance of this process. Explain the significance of this process. So we say meiosis produces haploid pollen and ovules with 10 chromosomes each. So the process of meiosis, like for example here we are saying 2 males breed each with 20. And let us use these diagrams of ours. We have, this is the female part of the maize. We have the ovary here, and then we have the style, and then we have the stigma. Now the ovary here is haploid. Our ovules here, which are going to be made, since the original parent has 20 chromosomes, the ovule, which is having the female gamete, will be reduced into a half. It will have 10 chromosomes. And here we are having our male part of the maize with the anther heads. Now these ones are the ones that are going to form pollen. Now the pollen which is also going to be formed here, it is coming from a parent who has 20 chromosomes. But the pollen will also be reduced to 10. So that is the process of meiosis. So we say meiosis produces haploid pollen and ovules with 10 chromosomes each. So what happens is that fertilization restores the diploid number to 20. So when we have our pollen grains moving from the anther and they go and pollinate the stigma, then we shall have the development of the pollen tube, which will take the female part to the ovule, the male part to the female ovule, with 10 chromosomes. And then also the ovule has also got 10 chromosomes. So after fertilization, we shall get a diploid zygote. 10 from the ovule and 10 from the pollen grain. When they combine, they will go back to the diploid number of 20. So we are saying that fertilization restores the diploid number to 20. And therefore, this one will ensure that chromosomes are stability within the species. This process of meiosis, which is taking place in the pollen and also in the ovule, is going to result in the maintenance of stability in the number of chromosomes in the new fruit or in the new offspring. We are still on the significance of meiosis. Item 3. This time we want to look at failure of meiosis. If meiosis fails, what happens? Learners are told that an error occurred during gamete formation, resulting in a gamete with an abnormal chromosome number. Resulting in a gamete with an abnormal chromosome number. So when we look at our diagram here, the diagram here is showing the normal process that is taking place. A diploid cell giving us haploid cell, the normal way how it is supposed to be. But in the abnormality, you may find that instead of having, for example, during this process, some of division may not take place. And here we may have no chromosome going here, and all the chromosome go here. Or we may have less going here and more going here. That is now what we are calling the abnormal chromosome number, which are going to exist in the chromosomes. Let's look at the tasks. When we look at the tasks, it says that state the role of normal meiosis in the chromosome number maintenance. State the role of normal meiosis in chromosome number maintenance. So, normal meiosis ensures accurate reduction of chromosome number by a half. So we are saying that in this abnormality, for example, if I have a cell here and it has got chromosomes, chromosome 1, it has got another chromosome here, it has another chromosome here, and it has another chromosome here. If these are homologous chromosomes, and this cell is undergoing cell division, and it forms two cells, we know that the number is going to be a half. So we expect this one to have two chromosomes at least. And then this one here, the second one, also to have two chromosomes. But when it comes that here we have 1, we have 2, and we have 3. And then the other one up, we have only 1 chromosome. Then now, that is what we are calling abnormality, abnormal chromosome number. So when this one happens, we say that meiosis has been abnormal. But in the normal process, this one will get 2 chromosomes, and the other one up will also get 2 chromosomes. That is the normal meiosis, and that is how it ensures accurate reduction in the chromosome number by half, such that this cell here has got a half, and this one has also got a half. That is normal meiosis. Predict one consequence if meiosis fails. They want us to predict at least one consequence if meiosis fails. So before we continue there, let us try to look at this karyotype here, showing us the number of chromosomes. We said that we have a diploid number of chromosomes in a normal organism, making it 46 chromosomes. Those are 23 pairs. So when we count the pairs here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. There are supposed to be 23 pairs of homologous chromosomes, which sums them up to be a total of 46 chromosomes. But when you look at chromosome pair 21, if you look at it, how many chromosomes are you seeing here? We are seeing 1, 2, 3. Meaning that there was a problem in meiosis. Meiosis failed. One of these chromosomes would have gone in another cell, but it ended up coming here. So here, instead of having 46 chromosomes, we have an extra chromosome, making it 47 chromosomes. So they are asking us to predict one consequence if meiosis fails. Now, if this is the chromosome representation of a cell, what consequence is likely to take place? So here we say failure of meiosis can result in what we call aneuploidy, leading to genetic disorders or non-viable offspring. So this whole condition here, this condition is called aneuploidy. Aneuploidy. When an extra chromosome is added to the total number of chromosomes, we call that one aneuploidy. And it can result in 2 genetic disorders or getting a non-viable offspring. So we say that this disrupts normal development and species continuity. So when such a thing happens, it is going to result in the abnormal development of the offspring, and the offspring may end up being non-viable, meaning that it is affecting the species continuity if the offspring is non-viable, and if there is disruption of development, that one is what we are referring to as the genetic disorder. We are still on significance of meiosis, item 4. This time we are looking at meiosis versus fertilization. Learners compare meiosis and fertilization in maintaining chromosome number. They are comparing meiosis and fertilization in maintaining chromosome number. So our diagram, what we are seeing here, this is a diagram of fertilization, whereby we are having a haploid male gamete with a total number of chromosomes 23, and we are having a haploid female gamete with a total number of 23 chromosomes. When these two come together and they are united, during a process called fertilization, the two have 23 chromosomes, and the 23 chromosomes of the different gametes, they combine together, and we get a total of 46 chromosomes in a diploid zygote. Let us look at task. Complete the comparison. So we want to complete the comparison here. And here we have got our diagram. Now this one represents meiosis. So we want to see what is taking place here. In meiosis, we start with a diploid parent cell, which is 2N. And here, in this cell, we have homologous chromosome. We have got one pair to represent the 23 pairs. In the homologous chromosome, we have the maternal chromosome, this one, and then the paternal chromosome. Whereas when you come this side of fertilization, we start with the haploid male gamete and haploid female gamete. Each one has got half the number. So these ones we call them N. But when you look at meiosis, when it is starting, these ones we are calling them 2N. So now here, what happens next during meiosis? Each chromosome duplicate, meaning that DNA synthesis takes place, forming 2 pairs of chromatid. So one chromosome from the mother is going to duplicate and it will form sister chromatids. And the one from the father will also duplicate and form what we are calling sister chromatids. That is what we are calling duplication. And that one is taking place during meiosis, that process which we call interphase. Now we come here in the first meiotic division, which we call meiosis 1. In meiosis 1, the homologous chromosomes are going to separate. Each one is going to go in its own cell. So in meiosis 1, we are going to end up with 2 daughter cells. Now, the parent chromosome of this homologous pair will go each in its own cell. That is meiosis 1. Now, when we are going to the second meiotic division, which we call meiosis 2, it is now the sister chromatids that are going to separate. But by the time we reach meiosis 1 and we separate the homologous chromosome, already now this one here, this one is haploid, and this one is also haploid. We no longer now call them 2N. Now this one is N, and this one also is N. They are half the number of this one. If you look at this cell here, this one which is the same as this one, look at this cell, it has got 2. Now when you look at this cell and this one, each one has got 1, meaning that this one and this one, they are half this cell. So these ones are haploid. By the end of meiosis 1, we have already halved the number of chromosomes. Now when we go to meiosis 2, what is going to happen here? It is just the breaking down of the chromosome so that the sister chromatids separate. So here we shall have breakdown of the centromere in the middle. This one will also break the centromere in the middle. And this one will go in this cell. And this sister chromatid will go in this cell. The same applies here. This sister chromatid will go in this cell. And this sister chromatid will go in this cell. And still, also these cells are still haploid. They are the same as these ones. They are half the number. But now when you look at this cell here, let us take and compare one cell, this cell here. Let's compare it with this one here. When you look at those 2 cells, you will see that this one has got 1 chromosome. And this one has got 2 chromosomes. So meaning that this one is already half the other one. So during meiosis ends up by producing a cell which is N. from a diploid cell which is 2A. That is meiosis. But when we come to fertilization, you can see here we are starting with a cell which is half the number of chromosomes, and another cell which is also half the number of chromosomes. So these two cells are going to combine. When they combine, they bring the chromosomes together and add them together, and they end up forming a cell which has got 46 chromosomes, which we are calling now a diploid. So when you look at fertilization, we start with half cells and we end up with a single cell with a diploid number. Half number ending up with diploid number. Whereas when it comes to meiosis, we start with a diploid number and we end up with a haploid number. So now let us complete the comparison. So here we have the process and the role in chromosome number maintenance. We start with meiosis. Meiosis, here we say, it reduces the chromosome number to haploid during gamete formation. What we have seen up here, from a diploid to haploid. Here we were represented by 2 chromosomes, and here we are having 1 chromosome. So when you see here 2 chromosomes, at the end we have 1 chromosome, that is meiosis. So that's what we are saying, that meiosis reduces the number of chromosomes to haploid, to a half, during gamete formation. But again, when we come to fertilization, if you look at fertilization, this one restores the diploid chromosome number in the zygote, meaning that during fertilization, the gametes which are haploid, like for example, we have here in fertilization, we have a haploid male gamete is combining with a haploid female gamete, each one with 23 pairs of chromosomes, and we end up with a diploid zygote, the end product, which has got 46 pairs, or 46 chromosomes, 46 chromosomes. So that's why we are saying that fertilization restores the diploid chromosome number in the zygote, whereas meiosis, it reduces the chromosome number to haploid during gamete formation. I hope you have understood that. We are still on significance of meiosis, item 5. This time we want to look at the species continuity. A conservationist explains why individuals of the same species always have the same chromosome number. Now, we are having our diagram here representing elephants of the same species, but they have variation. They look differently. But why is it that when they look differently, they still have the same number of chromosomes? So let's go to our task. Explain how meiosis contributes to this observation. They want us to explain how meiosis contributes to this observation. Which observation is that? They are looking differently. They are from the same species. Though they look differently, but still they have the same number of chromosomes. So how does meiosis contribute to this observation? So here we say meiosis ensures gametes have half the species chromosome number. So what is going to happen here does not matter. There are variation, but meiosis, what is going to do is to make sure that the gametes that are going to result into this individual and the gametes that results into this individual will always have half the number of the species chromosome number. And if at all fertilization restores the original diploid number. So meiosis, for it, what it is ensuring is to make sure that it has the species chromosome number. And fertilization, for it, what it does, it restores the original diploid number by bringing the haploid gametes together, each bringing half the number of chromosome, and then they become diploid number, going back to the original diploid number of the parent. State one significance of maintaining chromosome number in a species. They want us to state one significance of maintaining the chromosome number in a species. One significance, one importance of maintaining the chromosome number in a species. Now, this maintains the species identity and genetic stability. By maintaining the chromosome number, we are maintaining the species identity, meaning that this one will remain as an elephant from generation to generation. And also, it maintains the species genetic stability. It makes it to be stable without changes, very many changes, though variation may take place, but the chromosome number is genetically stabilized. Then also, it ensures proper gene expression and normal development. So when the chromosome number is maintained, there is going to be proper gene expression. Genes will express themselves properly, and then there will be normal development of the organisms in that particular species. Item 6, still under significance of meiosis. Let's now look at competence-based problem. A student suggests that mitosis alone is enough to maintain chromosome number across generation. Mitosis alone is enough to maintain chromosome number across the generation. So here we have our diagram or photo representing mitosis. I want us to look at this cell here. And you tell me the number of chromosomes that you are seeing. How many number of chromosomes are you seeing? This is the parent cell. So you can see that this is one, this is the second, this is the third, and this one is the fourth. There are 4 chromosomes. So it is undergoing mitosis. And when it undergoes mitosis, it gives us 2 daughter cells. And each daughter cell, now let us look at it. This is the last cell. If you look at this one, these are the last 2 daughter cells. Now I want us to look at these daughter cells. I want us to compare them with the parent. Compare the daughter cell with the parent. So when you look here, we say this one has got 1, 2, 3, 4 chromosomes. And when also you look at this one, it has got 1, 2, 3, and 4. So if you look here, the parent cell has got 4 chromosomes, and this one has also got 4 chromosomes. So the daughter is having 4 chromosomes, and even the parent is also having 4 chromosomes. That process of division is what we call mitosis. And mitosis results that a parent cell divides and gives us 2 daughter cells. And each daughter cell has got the same number of chromosomes like the parent. So here a student is suggesting that mitosis alone is enough to maintain the chromosome number across generation. Mitosis is enough to maintain the chromosome number from one generation to another. So let's look at our task. Evaluate that statement. They want us to evaluate this statement. Let us try to evaluate this statement and we see. Now this statement is incorrect. Why? Justify your answer scientifically. You have said the statement is incorrect. So can you justify your answer scientifically? Now we can say that mitosis maintains chromosome number within the body cells only. The process of mitosis is going to maintain the chromosome number within the body cell only, not in gametes, not in formation of gametes. It just maintains the chromosome number within the cell only. Without meiosis, fertilization would double chromosomes number every generation. So if mitosis would be the one responsible for formation of gametes, and the gametes that are formed, they have the same number of chromosomes like the parent number. Then when these gametes would combine together, then we were going to double the number. And the number would be around 92 chromosomes. The new organism will have 92 chromosomes, which is not viable. So it is only meiosis that during fertilization it will maintain the chromosome number, but without meiosis During fertilization, the number would be doubling. That is if we were using mitosis. Then also, meiosis. Meiosis is essential for maintaining the chromosome number in a sexual reproducing organism, whereby we are having sexual reproduction. Meiosis is very important in maintaining the chromosome number if we are having sexual reproducing organisms. So the learners by this time should be able to explain the role of meiosis in chromosome reduction. You should be able to link meiosis and fertilization to genetic continuity. You should be able to apply the knowledge to human, plants, and ecological context. And you should be able to analyze consequences of abnormal meiosis. And you should also be able to use correct biological terminology confidently. Thank you very much for participating in topic 11.1, which is inheritance. We have been looking at learning outcome A, understanding the process of cell division and significance of meiosis. So our learning activity has been to explain both verbally and in notes the significance of meiotic cell division in maintaining the chromosome number. Please visit me at Mugabi Joshua Biology. Don't forget to like, to share, and subscribe.